let's jump right into this explainer. We're going to nail down exactly how normal cells divide, and then look at what happens when those incredibly strict biological rules completely fall apart. We'll be pulling directly from Chapter 6 of the Form 4 biology textbook, and we'll apply it all to a really fascinating real-world scenario from Question 4 of the 2023 CADA Biology Trial Paper. Here's our roadmap for today. We're hitting the cell cycle and mitosis, meiosis and crossing over, abnormal division health issues, radiation and the cell cycle, and finally somatic versus germ cell mutations. Section 1. Cell cycle and mitosis. First, let's establish our baseline by looking at exactly how normal cells grow and divide.
The cell cycle operates in this beautifully strict, carefully controlled sequence. It kicks off with interphase, which is actually the longest part of the whole process. During the G1 phase, the cell grows and synthesizes proteins. Then we hit the critical S phase, where DNA synthesis happens and genetic material is replicated. After that, it's the G2 phase, where the cell gathers energy and makes its final arrangements. And once interphase is totally wrapped up, the cell jumps right into the M phase, which is where the actual physical division takes place.
Now, let's get precise with our textbook terms. Mitosis is specifically the division of the parent cell nucleus, a process we call karyokinesis. The ultimate goal? Producing two identical diploid daughter cells. That means every single new nucleus has the exact same number of chromosomes and the exact same genetic content as the parent cell. It's basically cellular cloning, and it's absolutely crucial for organism growth, tissue regeneration, and wound healing. Section 2. Meiosis and crossing over. Now let's shift gears from identical replication to the incredible variation of reproduction. Let's apply this straight to the 2023 CADA trial paper. There's a brilliant illustration of a key phase in meiosis here. Notice area X. This specifically highlights the homologous chromosomes pairing up during prophase I. When they pair up like this, they form what's known as a bivalent or a tetrad, simply because it consists of exactly 4 chromatids. But if you look at area Y, something highly dynamic is happening. The chromatids are actually overlapping at a specific point called a chiasma. This brings us right to a critical biological chain reaction that the exam question specifically asks us to explain.
So, how does this overlap translate to actual biology? Well, following the exact grading rubric for this CADA exam question, the process happening at area Y is called crossing over. Step 1, it causes an exchange of genetic material between non-sister, meaning non-identical chromatids. Step 2, this produces entirely new combinations of genes. Step 3, because of this, the resulting gametes will have totally different genetics. And finally, Step 4, this process directly causes variation within the same species. It's just an elegant molecular dance that literally drives biodiversity.
Section 3. Abnormal division health issues. So, we know the rules. But what actually happens when these strict cellular rules are broken? Well, the cell cycle is usually regulated by a special control system at the G1, S, G2, and M phases. When this control system fails, uncontrolled cell division takes over, forming a mass that we call a tumor. But not all tumors are the same. A benign tumor isn't inherently dangerous and can generally be removed surgically. A malignant tumor, on the other hand, is cancerous. It fiercely competes for nutrients, spreads through your blood and lymph vessels, and actively destroys the normal surrounding cells. Another massive breakage of the rules happens during meiosis. If the spindle fibers fail during anaphase 1 or anaphase 2, the homologous chromosomes or sister chromatids fail to separate. The textbook term for this failure is non-disjunction. Because of this uneven split, an abnormal gamete forms with 24 chromosomes instead of the usual 23. If that abnormal gamete is fertilized by a normal one, the resulting zygote will carry 47 chromosomes, meaning 3 copies of chromosome 21. This results in the genetic condition known as Down syndrome or trisomy 21.
Section 4. Radiation and the cell cycle. Now let's tackle a real-world application from the CADA paper, the intense effects of nuclear radiation.
Question 4 presents a pretty sobering scenario. It states, quote, "The Chernobyl nuclear accident occurred in 1986, releasing 5% of the radioactive reactor core into the environment. The paper then asks us to biologically explain the exact effect of this specific nuclear pollution on the cell cycle of the affected population." Let's trace how this builds based on the official answer scheme. The biological chain reaction is devastatingly direct. First, that radioactive core acts as a severe carcinogen or mutant. Second, this radiation directly disrupts the strict control system of the cell cycle across the G1, S, G2, or M phases. Third, without those built-in brakes,
uncontrolled cell division occurs. And finally, this runaway division forms a malignant tumor, cancer. It is a textbook tragic example of what happens when environmental factors break the cellular rules.
Section 5. Somatic versus germ cell mutations. Finally, let's tie it all together by looking at where these mutations actually occur within the body.
We are looking at two very different types of mutations here. Mutation X is occurring deep within the early development stage, affecting the embryo itself. Meanwhile, mutation Y is affecting a specific, localized area of an adult human body, like a skin cell or a muscle cell. As outlined in the marking scheme, the distinction between the two is massive. Mutation X involves a gamete, or germ cell. The absolute crucial takeaway here is that because it's in a germ cell, this mutation can be inherited from parent to child. Mutation Y, however, involves a somatic, or body cell. While it might cause major health issues for the individual, like localized cancer, it cannot be passed on to their descendants.
By taking this deep dive into the strict rules of cellular division and seeing exactly how radiation, mutations, and non-disjunction break those rules, we gain some really incredible insight. And it leaves us with a pretty powerful question. How might identifying and manipulating these microscopic mechanisms pave the way for the medical breakthroughs of tomorrow? I mean, could mastering the G phases or spindle fiber controls be the ultimate key to curing cancer? It's definitely something to think about. Thanks for joining me on this explainer, and keep questioning the science around you.